Hello again and welcome back to the second part of our lecture. So in our first lecture, we've discussed the cell theory and we discussed the classification of prokaryotes, their structures as well as their genetics. Now in this video, we would like to focus more on eukaryotes, their functions of their different membrane-bound organelles, discuss their cytoskeleton, and then finally go over the modes of the viral replication, discuss prions and viroids, and then as always conclude this video with some practice questions. Alright, so starting with eukaryotes, here I have provided you a summary of all the different structures that you can find inside a eukaryotic cell. And so as you can see, there are so many structures here that have membrane. Like for instance, the lysosome has a membrane, the peroxisome has a membrane, the mitochondria has a double membrane, the nucleus has a double membrane. So actually these are the characteristics that distinguish a eukaryotic cell from a prokaryotic cells, which includes one, presence of the membrane-bound organelles, like here nucleus and mitochondria are example of membrane-bound organelles, and second one is presence of mitotic or meiotic divisions versus binary fission that we see in prokaryotes. So here I would like to discuss the function of different one of these organelles, starting with the nucleus. So here you can see that we have the nucleus that has the double membrane, and then we have the nucleolus that is located inside. So let's start the writing here. So the nucleus is basically the storehouse for the genetic material, where it serves as a storage place for DNA. And then it's inside the nucleus that DNA can undergo replication, or it can undergo transcription where mRNA will be formed, and then later on this mRNA with the help of these nuclear pores can exit the cell and go reach the ribosomes, which are shown here in red, and undergo translation, and by translation proteins will be made. Okay, so going back here. So we just discussed nucleus is the storehouse for DNA. There are histones that will help compact DNA into the condensed form called chromatin. And then during cell division, this chromatin will further undergo coiling and form chromosomes, which is a very condensed form of DNA and proteins. The other function of the nucleus is that it has a nuclear envelope that has pores inside it. So it's basically perforated, and as we just shown it, it's called nuclear pores, and the function of these nuclear pores to allow movement of large molecules. So there are nuclear pores allow movement of large molecules, like for instance mRNA or the ribosomal subunits. So in fact, the fourth function is that inside the nucleus we have nucleolus, which is the location where the ribosomal RNA and ribosomal subunits, as we just discussed, 40S and 60S in eukaryotic cells, so ribosomal subunits are being produced. So our RNA and ribosomal subunits are being produced here in the nucleolus, and then with the help of these nuclear pores, these structures can exit the nucleus, and then once inside the cytoplasm, they could assemble to form the ribosomes, which are shown here in red. Alright, the next structure I would like to discuss is the mitochondria. And so here you can see that the mitochondria is made of a double membrane, and this inner membrane is kind of like finger shaped. It keeps on having like convoluted structures. The reason for that is to increase the surface area. So let's go over that. So the mitochondria is basically a bean-shaped structure, and its job is to supply ATP. So we do have a lecture on glycolysis, anaerobic respiration, and aerobic respiration. We have extensively discussed the function of every one of these structures. complex 1, complex 2, 3, and 4 of the electron transport chain will create a hydrogen gradient, which will later on use by the ATP synthase to generate ATP. And this is called aerobic respiration. So I'm not going to go through details in this video. I would just like to focus on the structure of mitochondria. So the function of mitochondria is again to supply ATP. It's bound by a double membrane and then the inner membrane. So here I just showed you that this is the structure of the mitochondria, and then the inner membrane has many folds, and these folds are actually called cristae. So there are many folds that are called cristae, and the function of these cristae is to increase the surface area of the inner membrane. And the reason for that is that it's actually inside the inner membrane. So here you see we have the inner membrane right here, then we have the outer membrane that is right here. So in this image that I've shown, this is the inner membrane that forms the cristae, and then the outside one is the outer membrane. The space between the outer and inner membrane is called intermembrane space. That's where the proton gradient is being formed. And then finally, the space inside confines of the inner membrane is called mitochondrial matrix. Now back to the first topic that I just discussed, the inner membrane has so many cristae to increase surface area, and the reason for that is that the electron transport chain, which are all these complexes that I've shown you here, is located on the inner membrane. So we want to incorporate as many electron transport chains as we can to generate a lot of energy, because if you think of it, imagine if the mitochondria had just a smooth inner membrane. How many electron transport chains could be had there? Probably not that many. Versus in this situation, look how many we can have. Because of the increased surface area, we can have many electron transport chains, which will increase its ATP production. Okay, and then certain features of mitochondria that give clues that these are probably simple prokaryotic cells that millions of years ago were engulfed by eukaryotic cells is that mitochondria is composed of 70S ribosomes. Okay, so remember 70S ribosomes was in prokaryotes. So mitochondria also has its own ribosomes to produce its own proteins. So it has 70S ribosomes. It's self-replicating. And indeed, if it replicates in a manner similar to binary fission. Okay, and so again, that's another feature that we see in prokaryotic cells. The next two structures that I would like to discuss are ribosomes and the endoplasmic reticulum. So here we have the ribosomes. We just discussed these red structures. These are for protein production. We have ribosomes that are by themselves in the cytoplasm, and the products of these ribosomes is usually staying inside the cytoplasm, versus there are ribosomes that are, some of them are bound to the endoplasmic reticulum. So here you can see all these membrane structures that have the red dots, which are ribosomes attached to it. So a ribosome that is attached to endoplasmic reticulum is called rough endoplasmic reticulum, versus the ones that don't have it are called smooth endoplasmic reticulum. So the function of the ribosomes that are bound to the rough endoplasmic reticulum is usually for the protein that are destined for excretion outside the cell. Because if you think of it, now the protein that is formed in the rough endoplasmic reticulum can go to the Golgi apparatus, and Golgi apparatus has the capability to secrete these proteins as a vesicle. So again, ribosomes that are inside the cell usually produce proteins that stay inside the cell, versus the ones that are on rough endoplasmic reticulum produce proteins that are being secreted by secretory vesicles. Okay, let's go back now to the table and discuss. All right, so endoplasmic reticulum is basically an interconnected network of thin folded membranes. There are two forms called rough endoplasmic reticulum, which contains ribosomes, and therefore the proteins that are being produced by the rough endoplasmic reticulum are often destined for the cell membrane or for secretion outside the cell. And then one other important feature I would like you to know here is that rough endoplasmic reticulum is continuous with the nuclear membrane. All right, so nuclear membrane can be continuous with the rough endoplasmic reticulum, versus the smooth endoplasmic reticulum, which extends from the rough endoplasmic reticulum, and its function is to produce steroids, fatty acids, and to detoxify drugs or toxins. And then we just discussed the difference between smooth and rough endoplasmic reticulum is that smooth does not have ribosomes. Okay, we briefly touched upon the function of the Golgi apparatus. Basically, Golgi apparatus is consisted of 4 to 6 stacks of membrane enclosed spaces. So here you can see that we have the Golgi apparatus that should be made of 4 to 6 stacks of membranes. The area that receives the vesicles is called cis, and the area that the vesicles exit are called trans. So the cis or entry face of the Golgi apparatus, it usually faces the rough endoplasmic reticulum because it wants to receive all the proteins and vesicles from rough endoplasmic reticulum. And then the side where vesicles exit are called trans or exit, and this side is usually facing the cell membrane. So we have the rough endoplasmic reticulum usually on this side, and then cell membrane is usually faced on the trans side. So if we refer back to the image I've shown you here, you can see that here we have the cis or entry side is facing the rough endoplasmic reticulum versus the trans side that is facing the cell membrane, and that's the site where the vesicles will exit and get excreted as secretory vesicle. All right, the structure that I missed last time, it was lysosome, which is shown right here, and you can see that there are different cellular structures or debris or whatever from outside that has been picked up is being digested inside the lysosomes. So all these structures that I'm showing here is basically like a food that is being digested inside the lysosome. And then finally we have peroxisomes that is shown right here. So now let's go over the function of these two structures. So lysosomes, let me pick a different color, there we go. So lysosome, these are membrane-bound vesicles that contain digestive enzymes. And these digestive enzymes can help either break down the damaged organelles. So if that happens, it's called autophagy, because now the cell is digesting its own old organelles. Like for instance, if there is a damaged mitochondria, lysosome will start to eat that damaged mitochondria, and that's called autophagy. Or it can help defend against pathogens, and so therefore any particle that enters the cell, the lysosome can engulf them and start digesting them. And so what happens after these molecules get digested is that the broken down product is being released back by the cytoplasm, and then the cell will use those broken products for other purposes. And then finally we have peroxisomes. These are again membrane-bound vesicles, and these peroxisomes contain oxidase enzymes. Oxidase enzymes, which are used for breakdown of long chain fatty acids through the process that is called beta oxidation. So beta oxidation is the process through which long fatty acids are being broken down in the peroxisomes. And then there is usually H2O2 or hydrogen peroxide that is being produced as a byproduct of these reactions. And in order to defend against H2O2, because H2O2 is a reactive oxygen species, peroxisomes contain the enzyme called catalase. that will break down H2O2 into H2O plus O2. All right. Now next I'd like to discuss the cytoskeleton of the eukaryotic cells. So there are three important components that you should be familiar with, and these include the microfilaments, microtubules, and intermediate filaments. So microfilaments, these are actually the smallest form of these fibers. So this is the smallest form of these fibers that allows the cell to move around and divide. It also plays an important role in muscle contraction and provides support to cell and its microvilli structures. Microtubules, on the other hand, these are long tubes that are made, as the name implies, tubules that are made of tubulin, and their function is to help maintain the shape of the cells and help with the movements of organelles. So you know how during cell divisions there are spindle fibers. These spindle fibers are made of microtubules that attach to the chromosomes and pull them apart. So that's the job of the microtubules, to help move organelles around. In addition, microtubules are found in the structures of cilia and flagella. So they are found in cilia and flagella, the function of which is to help with the movement of cells. And then finally, we have intermediate filaments, and these are somewhat smaller than microtubules, but then they are bigger than microfilaments. That's why the name intermediate filaments are given to them. Again, their function is to provide support to cells and then help anchor organelles such as nucleus. The next structures are centrosome and centriole. So basically centrosome, it's a dense region of the cytoplasm that contains two smaller structures called centrioles. So here we have a centrosome that is made of two centrioles. And then the centrioles themselves are composed of short microtubules. So all these tubes that you see right here, these are microtubules. And then these centrioles are perpendicular to each other. So it's not just for the drawing purposes that you see them perpendicular. They are arranged perpendicular so that the fibers that are extended can extend in different directions, perpendicular to each other. And then one other important feature that I would like you to know is that in a centrosome, the centrioles have a 9 plus 0 triplet pattern. And what I mean by 9 plus 0 triplet patterns is that we have 9 of the triplet microtubules. So here we have 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 triplets. And since there is none at the center, we call it 9 plus 0. Versus in microtubules that you see in cilia or flagella, they have a 9 plus 2 pattern where there is a 9 doublets of microtubules. So here 1, 2, 3, 4, 5, 6, 7, 8, 9 doublets of microtubules, and then there is also 2 central microtubules, 1 and 2. That's why the name 9 plus 2 pattern is given for the microtubules that you find inside the cilia and flagella. So now, speaking of cilia and flagella, let's focus a little more on the functions of these structures. So basically, cilia are hair-like structures that are being projected from the cell surface, and their function is usually to help with the movement of fluids and material across the cell, and they also can help with the cell movement. And one a good example is, for instance, the cilia that you will find on the respiratory tract. They help move the dust upward so that we can cough them out. So cilia will be helping with moving the material around, or movement of fluid, versus flagella, which is longer than the cilia and has kind of like a whip-like appearance, and its main function is to help the cell to move around. And as we just discussed, like sperm has a flagella, and the function of its flagella is to help it move around. All right, next I would like to discuss the viruses. So as we just discussed earlier, viruses are having their own genetic material, but since they cannot divide on their own, they have to be relying on a host cell to divide. So therefore, since they cannot divide on their own, they're not considered as a living organism. Now, the common structures that you will find in all viruses. So here I have shown you different forms of viruses. Bacteriophages are the viruses that only infect bacteria. You can see that they have a head, they have a neck, whiskers, there's a tail structure, and then finally they have tail fibers that help them land on the bacterial cell. Then we have non-enveloped viruses, where you can see since it doesn't have a membrane around it, it's called a non-enveloped virus. Okay, and then those that have a membrane around it are called enveloped virus. So the features that you should be familiar with that you will find in these viruses include nucleic acids. So you can see that all these three viruses have nucleic acid inside the structure that is called capsid or protein coat. And this nucleic acid could include RNA or it could be DNA. All right, and then again, this RNA and DNA can be in different forms. They could be double-stranded, like you could have double-stranded RNA, you could have double-stranded DNA, you could have single-stranded RNA or single-stranded DNA. These could be circular or they could be linear. So they come in variety of shapes and forms. But in any case, they are all nucleic acid and are made of either RNA and DNA. The next structure that you will find in these viruses is called capsid, which is a protein sheet that will protect the nucleic acids. And then sometimes the capsid itself could be enveloped by a membrane. And this membrane, as you can see here, or envelope, this is derived from the host cell. So when the cell is lysed and the particles are released, they pick up some of the membrane from the cell and they use it to enclose their capsid. And so that's the difference between enveloped viruses and non-enveloped viruses. Non-enveloped viruses have only a capsid around their nucleic material, versus enveloped viruses have a membrane that is surrounding the capsid. Now, in terms of the replication, basically each virus can replicate in a limited number of hosts. Like for instance, bacteriophages can only infect bacteria. They cannot infect human cells. Or HIV virus, for instance, it can infect human T lymphocytes, but it cannot infect bacteria. So therefore, that's what I mean by each type of virus has a limited number of hosts. Viruses also lack ribosomes, and so therefore they don't have their own protein synthesis machinery. So the genes that they have will actually recruit the host cells to use their ribosomes to replicate viral components, and then there would be assembly of these components, and then the new viral components will be released as viruses. So again, viruses, they have the genetic material to recruit and basically enslave the cell to make their products, but they don't have their own ribosome to produce their own proteins. Okay, so that being said, let's go over the life cycle of a bacteriophage, which is a virus that infects bacteria. So there are two phases to the bacteriophage life cycle, and this include the lytic phase, where virus particles are being produced, assembled, and it causes lysis of the bacterial cell, and then the viruses will infect again another cell. Versus it could exist in the lysogenic cycle, where the DNA will incorporate into the bacterial genome and it stays there dormant for a few generations until eventually it decides to come out and then starts infecting the cells again. So therefore, bacteriophage replication cycle can exist as either lysogenic or lytic phase. And so let me write the description here for you. So during the lytic phase, bacteriophage plasmid will produce proteins like, for instance, the capsids, and also new DNA. And once these are assembled together, there would be new viral particles that will break open, or in other words, lyse the cell. And these viruses can now go and infect more bacterial cell. That's why this is called a lytic cell, because the cell have lysed, or in other words, has been broken open. And then the next phase is the lysogenic phase, during which time the phage or the bacteriophage genome is incorporated into host DNA. All right, but it doesn't produce any proteins. It's just incorporated there. So anytime the bacteria is replicating its DNA and is dividing, so does the bacteriophage DNA. So it keeps on being transmitted to the next generation bacteria. But then during this phase that the phage genome is incorporated into host DNA, there will not be any lysis of the bacteria. The bacteria will not lyse, and the host cell can continue to survive. At some point, eventually, this DNA will come out as a plasmid and start lysing the cell. So there could be conversion of lytic cycle to the lysogenic cycle, or backward, lysogenic cycle converting into the lytic cycle. All right, next is a hot topic that is commonly asked on MCAT, and that is retroviruses, an example of which includes, for instance, the HIV virus, or the other name is human immunodeficiency virus. Once it starts to kill too many of human T lymphocytes, and the human T lymphocytes drops below a certain number that we know that that person is so sick, that's the time that we refer to the infection as AIDS, acquired immunodeficiency syndrome. Okay, so AIDS is the advanced form of HIV, meaning that already the immune system of that person has started to fall apart. Now, since this is a hot topic, I would like to discuss a little more detail than the rest of the viruses regarding how does HIV replicate and what are the components of HIV. So basically what HIV does is that it attacks the human CD4 positive, which are helper T cells. And the function of helper T cells is to activate other B cells in order to make them produce antibodies and so on. So basically HIV is targeting our immune system. As a consequence of which, once our immune system is weakened, therefore now any opportunistic infection can cause death in these individuals. Like a common cold, our body can't fight it anymore and can kill it. So basically that's how HIV works. It's not the virus that directly attacks and kills us, but rather the virus weakens our immune system, and now any other opportunistic infection can attack us and kill us. Now a common question that is asked is, why can't we really find a solution to HIV? Why can't we find an antiviral medication that attacks it? And the answer is because the genome of HIV is made of a single-stranded RNA. And RNA is very prone to developing mutations. Unlike DNA that is double-stranded and has a very solid and stable structure, this single-stranded RNA is prone to developing mutations. So the structure of the HIV virus is continuously changing. And so once you throw a medication on the HIV virus, it won't be long before there's a new mutation that the HIV virus is now immune to it. So that's why patients who have HIV, there are multiple medications that are given to them at the same time in order to restrict the HIV replication. And the patient is always told that if you start to take the medications off and on, it's very likely that the virus will develop resistance. You cannot do that. Even if you take it on time, you can develop resistance. But if you take it off and on, then there is a higher chance, because now the virus can replicate and there are more of them, and they have a better chance of developing new mutation that is resistant to all the different forms of medication that we are giving these HIV patients. But in any case, that's the reason we give multiple antiviral medications to HIV patients in order to restrict the virus as much as we can. But if the patient is non-compliant and take the medication off and on, then that's the time that there is a higher risk of developing resistance. Now, in terms of the components of the HIV virus itself, HIV virus has 3 genes, and these genes include the GAG gene, the function of which is to produce viral structural proteins. It has the POL gene, the function of which is to produce reverse transcriptase, as well as the integrase enzyme. And then again, that's another hot topic that is being asked on MCAT, because HIV has the RNA genome. So how does it incorporate its genome into the host DNA? Because after HIV infects, we know that it can stay dormant for many years before it becomes active. And during the time that its DNA is incorporated into our genome, that's the time it's dormant. But how did it incorporate RNA information into our DNA genome? And the answer is via the reverse transcriptase enzyme. So reverse transcriptase enzyme converts RNA into DNA. So we know how the genetic material is usually flowing from DNA. Through transcription, it becomes RNA. And then through translation, a protein is being formed. So this is called transcription. In HIV virus, however, RNA is being converted to DNA. Right, so let me pick a different color. RNA is being converted to DNA. That's why we call it reverse transcription. And this is being done with the help of the reverse transcriptase enzyme. And then once the DNA is formed, then the integrase enzyme helps to integrate DNA of the virus into our genome. It can stay there for many years until, for whatever reason, the virus decides to go active again. It starts to form new viral particles that start attacking our human CD4 T lymphocytes, and that's the time that it can advance and form AIDS. Okay, so keep that in mind regarding the function of the reverse transcriptase and integrase enzyme. And then the final gene is called ENV gene, which is forming the viral envelope protein. Okay, so here you can see that we have a T lymphocytes. It has the CD4 receptors on its surface, and then there are also co-receptors called CCR5 or CXCR4 that are there. So the HIV virus recognizes these receptors, enters, and then can integrate its DNA into the genome, and then later on become active and form new viruses. And so the fact that there are co-receptors here, that's the reason that some individuals are more resistant, or some of them are actually immune to HIV virus, because there has been mutation in their co-receptors, and therefore HIV virus cannot recognize their CD4 T lymphocytes. So again, mutation in the co-receptors is the reason that some humans are very resistant or even immune to the HIV virus. Okay, so and then from here on, we just discussed, once the HIV becomes active, it starts attacking T lymphocytes. If the CD4 counts drops below 200 cells per millimeter cube, that's the time that we are having the advanced form of infection that is referred to as AIDS or acquired immunodeficiency syndrome. All right, the next topic are prions and viroids. So first I would like to describe the viroids. So basically viroids, these are subviral particles that are made of a circular RNA, and they do not have a capsid. Okay, and viroids are usually most infectious in plants. They are found in plants. So once again, viroids, they have circular RNA, and unlike viruses, they do not have capsids, and these viroids are mostly infectious in plants. Next, we have prions. So let me erase all this. Okay, next we have prions, which are the proteinaceous infective particles. So these are actually proteins, and they do not contain any nucleic acids. Okay, so unlike viruses, prokaryotes, viroids, the prions is the only example here that does not have any nucleic acid materials. And you might have heard of the mad cow disease. So basically prions attack the brain. And so that's where the mad cow disease has come from. It's from prions, which are infectious proteins. So now let's go over the mechanism of spread of prions. So here what you can see is called a cellular prion protein, or PRP. This is actually a healthy protein. This is a normal protein that is found in healthy individuals. This protein is sensitive to the protease. It has alpha helical structure, and since it's sensitive to protease, it could get digested. However, there could be mutation in this PRPC protein, as a consequence of which they could get converted into a protein called PRPSC, which is shown right here. So there could be spontaneous generation of PRPSC. And these PRPSC proteins have a beta sheet protein structure and are resistant to proteases, so the body can no longer destroy them. And since these proteins will attack the brain, they can cause spongiform encephalopathy, which is a human version of mad cow disease. So spontaneous mutations is one mechanism of generation of the PRPSC proteins. Okay. The other mechanism is, however, having the PRPSC protein coming in contact with these normal PRPCs, and upon their contact there would be conversion of the normal proteins into the mutated form, or PRPSC. Okay, so once again, there are two different modes of converting PRPC normal protein into PRPSC structure, and that is one by mutations, and second one upon contact of one of the normal proteins with the abnormal one. And so that's the reason that if a human consumes meat products that already contain this PRPSC protein, they can now have the infectious prion that will increase in number and cause the spongiform encephalopathy, the other name for which, so I just wrote it, spongiform encephalopathy. The other name for which is Creutzfeldt-Jakob disease, or CJD. And so that's the reason that once they find any animal like a deer or a cow with a mad cow disease, right away they kill those animals and they burn it, because we don't want these infectious proteins to spread around or get infecting any of the humans. All right, now there are some questions that I would like to answer here before I move on with the practice questions. So I didn't explain what spores are. Do you know what are spores that are found in bacteria? And so basically these spores are made of dipicolinic acid with a keratin-like coat, and this structure makes these bacteria extremely resistant to chemicals and heat. So you might recall that there has been some biologically terrorism by sending out letters that had the Bacillus anthrax, which was a type of bacteria that had spores. So everyone was warned that if you even iron those envelopes, it won't kill the bacteria, because the bacteria had spores and they're extremely resistant to heat. But there is a way to kill them, of course, if you autoclave them, which is a combination of heat in a moist environment that still kills the bacteria. But my point is, the function of spores is to provide a lot of resistance to chemicals and heat. The next question that I have here for you is, what are cyanobacteria? So basically, cyanobacteria are referred to the bacteria that can undergo photosynthesis. So they can photosynthesize, which means that they will produce carbohydrates from solar energy. Now, what are chloroplasts? So these are the organelles that you will find in algae or in green plants. And their function is to, again, let these cells undergo photosynthesis, which means that they will use solar energy to convert CO2 and water into carbohydrates, like for instance glucose, and oxygen. So basically, photosynthesis is the reason that these green plants can pick up CO2 and release O2, because they will combine CO2 with water to give out sugar, and then the O2 will be released back into the environment. Now, the next question is, what's the difference between euchromatin and heterochromatin? So euchromatin is a lightly packed form of chromosome that is transcriptionally active, versus heterochromatin, it's a tightly packed chromosome that is transcriptionally inactive. And examples of heterochromatin include, for instance, centromere or telomere. So if you recall, having a chromosome like this, the ends of the chromosomes are called telomeres, while the center is called right here, the centromere, which is the site of attachment of the microtubules during cell division. And to be more specific, it's the kinetochore site, and the centromere is where the spindles attach to. The next question, how did eukaryote evolve from prokaryotic cells? So we all know that the very first cell that most likely developed was a prokaryote, but how did it evolve into eukaryote? So it's thought, again, everything here is a thought, we can't tell for sure, but it's thought that there has been invaginations in the plasma membrane that had led to the creation of the nuclear envelope. So now we are having a cell that has a nuclear envelope that can store the genetic material. And then later on, through the endosymbiotic relationship, some of these cells have picked up bacteria that had, for instance, the aerobic respiration ability. And these bacteria that has been picked up has turned into the mitochondria. So they were picked up here and they became mitochondria. And so now their function is to use oxygen to generate ATP for this eukaryotic cell. Now there are multiple evidences that supports that the mitochondria has been most likely a prokaryotic cell, and I've already touched on some of the evidences, but I would like to review them with you one more time. So number one, mitochondria and chloroplasts, both of them have a similar size and shape to bacteria. Okay, so size and shape of mitochondria and chloroplasts are both similar to bacteria. They also both contain their own DNA and divide independent from the rest of the cell in a binary fission fashion. They also contain S70S ribosomes. They do not have any introns, and similar to bacteria that once they transcribe their mRNA, there is no mRNA processing. So in eukaryotes, mRNA still needs to have 5' capping. It needs 3' end polyadenylation, and then its introns, which are the non-coding regions, has to be taken out via the splicing process. None of this happens in bacteria nor in mitochondria or chloroplasts, because one, they don't have any introns, And 2, they're not undergoing any post-transcriptional modification. There is no 5' capping or polyadenylation. So all these evidences suggest that most likely the mitochondria, so mitochondria and chloroplast were prokaryotic cells that have been engulfed by larger eukaryotic cells. All right, so now we can move on to some practice questions. If you need to take a few minutes break, then by all means you can go ahead and pause the video. Otherwise, I'm going to read the questions for you and then go over through the answers. So, which of the following features does not distinguish eukaryotes from prokaryotes? Okay, next question. Which of the following is the site of the rRNA production? The next question is, which of the following structures are present in bacteria? Then we have, which of the following cells contain 70S ribosomes? We just discussed it. And then, which of the following bacterial structures is analogous to eukaryotic transport chain? And then finally, which of the following structures consist of a double membrane? All right, so if you need more time, then you can go ahead and pause this video. Otherwise, I'd like to start with the first question here. Which of the following features does not distinguish eukaryotic from prokaryotic? So, presence of the membrane-bound organelles. That does distinguish eukaryotes from prokaryotes because eukaryotes have membrane-bound organelles while prokaryotes don't have it. Presence of lysosome, again, that's the same thing. Lysosome is a membrane-bound organelle. Presence of genetic material, well, both eukaryote and prokaryote have genetic material, so this choice does not really distinguish these two cells from each other. And then mitotic divisions, again, that also distinguishes eukaryote from prokaryotes because we discussed eukaryotes undergo mitosis, and then the germline cells undergo meiosis, while prokaryotes undergo binary fission. The next question is, which of the following is the site of the rRNA production? So you recall we just discussed how if this is the cell, then we have the nucleus, and inside the nucleus we have nucleolus. And then nucleolus is the site of the rRNA and ribosomal subunit production. These products will exit the nucleus via the nuclear pores, and then it's inside the cytoplasm that they would assemble to form ribosomes. Okay, so the answer is nucleolus is the site of the rRNA production. Moving on to the next question. Which of the following structures are present in bacteria? Histones is not present. We mentioned that histones and introns are only specific to eukaryotes. Mitochondria is a membrane-bound organelle. You don't see that in bacteria. Nucleus is also a membrane-bound organelle. You don't see that. So the answer is ribosome. And if you refer to the previous lecture that I gave you, I had two circles that shows the similarities between bacteria, between prokaryotes and eukaryotes, and ribosomes was right in the middle because ribosome is found in both prokaryotes and eukaryotes. The next question is, which of the following contains 70S ribosomes? So without looking at the answer, I know that 70S ribosomes is found in prokaryotic cells like bacteria, it's found in mitochondria, and it's found in chloroplast. Now with that information in mind, now I can go back and read these questions. Drosophila gut cells, no, these are eukaryotic cells, so this is incorrect. Human intestinal bacteria, we are talking about bacteria, and so yes, that is correct. Mammalian mitochondria, yes, mitochondria is the correct answer. And then plants chloroplast, that is also correct. So the answer would be 2, 3, and 4. Choice D. Then we have the next question which says, which of the following bacterial structures is analogous to eukaryotic electron transport chain. So remember, the electron transport chain is found inside the inner membrane. Okay, so it's actually a membrane that contains the electron transport chain. So therefore, the plasma membrane of bacterial structures is most similar to what we have inside mitochondria. And then the next question is, which of the following structures consist of a double membrane? We already discussed this. Mitochondria has a double membrane. Nucleus also has a double membrane. Endoplasmic reticulum and Golgi body, you can see how they are like overlapping like this. So you can also consider them as having a double membrane. And let me show you the image right here. So you can see the nucleus has a double membrane. The mitochondria has a double membrane. Here you can also, these vesicles, you can consider them as a double membrane. So again, it depends on the examiner whether they want to refer to them as vesicles or double membranes. But here, since you have two membranes that is right adjacent to each other, you can consider them as double membranes. Again, I reviewed many different books. Some say yes, there are double membranes. Some say no, these are vesicles. So it depends which book you're referring to, but my point is that these membranes are coming very close to each other. Okay, so definitely mitochondria and nucleus, and after reviewing different books, I've noticed that they're also referring these structures as double membranes. So we can say that all these can have two membranes that are adjacent to each other. All right, next set of questions. Which of the following is or are the primary functions of a capsule? And the next question is, how do bacteria move towards attractants? What is the definition of the bacterial conjugation? And then which of the following is true regarding DNA division during binary fission? Okay, so if you need to pause the video, you can do that. The first question is, which of the following is the primary function of capsule? So we just described how the function of capsule is to protect bacteria against drying. It can prevent phagocytosis, and then it can also help with attachment to different surfaces. That's another function that I didn't mention in my previous lecture, but that's also important. So of all the choices that is listed here, protecting against phagocytosis, that would be the correct answer here. So choice A would be the correct answer. Moving on to the next question. How do bacteria move towards attractants? So again, without looking at the answer, if there is a counterclockwise motility, then it's moving forward. All right, so that's counterclockwise. And then if there is clockwise motility, then there is tumbling motion. Okay, so if a cell is moving towards attractant, we want it more to have moving forward, or in other words, to be in a running mode. So how do they move towards attractants? By increasing their counterclockwise motility, so counterclockwise rotation, to promote a running motility. So choice C would be the correct answer. Next, what is the definition of bacterial conjugation? So we just discussed transformation is when DNA is picked up from extracellular environment. Transduction is when a bacteriophage helps transfer gene from one bacterium to another. And then finally, conjugation was when a sex pili was formed to help transfer plasmid from one cell to another. So here it would be gene transfer by sex pili. That would be the definition of conjugation. And then the next question is, which of the following is true regarding DNA division during binary fission? Okay, so DNA size of daughter cell is half the parental cell. No, we just have shown in the previous lecture that the DNA would be identical. Multiple origins of replication are used for replication. That is true for eukaryotic cells. We just discussed bacterial cells have a circular DNA, and there would be a single origin of replication, and replication will progress bidirectionally from both sides. DNA replication is semi-conservative. That's true, because what it means is that of the parental cells, one of these circular DNAs is from the original parent, and then the newly synthesized one is also there. So it's semi-conservative model, meaning that one of them is from parents and then the other one is new. That's why it's called semi-conservative. Okay, so choice C is correct. And then regarding choice D, both daughter cells acquire their genome from a single strand of parental DNA. No, it's a double-stranded DNA. Okay, double-stranded circular DNA is more accurate to say. So this is also incorrect. Now the last set of questions that we have here is, which of the following processes produce two daughter cells that are genetically identical to parental cells? Then we have, which of the following is the correct order for gram staining? And then the last question is, which of the following processes of gene transfer can transpire in both eukaryotes and prokaryotes? Okay, so the first question is, which of the following produce two daughter cells that are genetically identical? So mitosis and binary fission produce genetically identical daughter cells. Meiosis, however, a 2N, remember, was being converted into haploid cells. So it's not genetically identical. Furthermore, there is crossover that would increase genetic diversity of their products. So meiosis is not producing identical products. So the answer would be 1 and 3, or choice B only. Then we have the next question, which says, which of the following is the correct order of gram staining? So without looking at the answer, remember we had crystal violet in order to stain the peptidoglycan cell wall. Then after that we had the iodine staining to strengthen the crystal violet staining. After that we would wash with ethanol so that any excess crystal violet would be washed off. The gram-negative bacteria would lose all their staining because they have a really thin peptidoglycan cell wall. And then after that, we use the safranin counterstain in order to stain the gram-negative bacteria. So here I have a summary for you again. Right? So we have crystal violet, then iodine after that. That stains all the gram-positive bacteria. Alcohol then to decolorize the gram-negative ones, and then finally safranin is for providing a reddish counterstain to gram-negative bacteria. So therefore, now looking at these answer choices, choice B would be the correct answer. Crystal violet, iodine, ethanol, and safranin. Alright, and then the final question that we have here is, which of the following processes which of the following processes of gene transfer can transpire in both eukaryotic and prokaryotic cells? So we just discussed how transformation, conjugation, and transduction, these are the modes of horizontal gene transfer that happens only in prokaryotic cells. Transposition, on the other hand, which was having a piece of DNA coming off from one site and then moving to the other one, that can happen in both type of cells, in both eukaryotic cells and prokaryotic cells. So therefore choice D would be the correct answer for here. Alright, so that concludes our discussion of this video. Thank you all for listening. Of course, there is more practice questions that you can always take in our QBank. There are passage-based questions there as well. But after listening to this video, I hope you feel now more comfortable with tackling any one of these topics. Again, thanks for listening, and see you for the next video.